thank you very much for this invitation. I'd like to be here in Liège. It's a pleasure to share with you a few days to speak about, in my case, things related to biology, but actually my talk will be more of a warning. I realize that this is the first talk about biology in this school, but I think this is important. I will talk a little bit about biology, but I would mostly talk about misconceptions or a priori ideas that we in general, human beings and many disciplines, including biology, have sometimes about biological evolution. And I think it is very important to set up basic ideas about how evolution works. And first of all, and precisely relating to, there are many kinds of misconceptions, but I will focus mostly on things that relate to the origin and evolution of life and astrobiology issues, if you want. But first of all, how does evolution work? That is something very important, very simple at the same time, but that can trigger or lead to a variety of misunderstanding. So evolution is a process that works by via two types of underlying mechanisms. The most very well known is something very easy to understand. Is a mechanism that encompasses two sequential kinds of mechanisms. The first involves reproduction with variation, so you have individuals that reproduce and give birth to an offspring which contains variation. And so this is the first step, reproduction plus variation, and then you have natural selection that can act on this variation and eliminate the variants that are less fit. And of course, natural selection has to do with the environmental selection. So the environment is key, is very important here. So imagine that in this particular environment there is a predator that that represents a selective force that acts against many members of this population here and just removes all the variants that are less fit. In this particular case, all those variants that are not hidden by any particular kind of adaptation. We call this particular feature adaptation. So this guy is adapted to live in this kind of environment and thus escape this kind of predator. And then the immediate consequence is that this guy here will now be able to reproduce and give again rise to a variety of offspring with variants that are perhaps more similar to what he looks like, because it is adaptive in this particular environmental context. So this is very important, very simple, but very important to understand. So these kind of mechanisms, we call them Darwinian evolution. It means that there is reproduction with variation upon which natural selection in given environmental conditions can act and just select for the most fit individuals. Something very simple. There is a second kind of mechanisms that can also give rise to evolution, and that is the following. You have an individual that reproduces the same scheme as before, but here maybe you don't need these kind of mechanisms don't necessarily imply that is natural selection acting and removing organisms from a given population that are less fit, but simply that there is some what people call neutral processes or stochastic processes that lead to some particular individuals to reproduce in a given time and space. So this is what we can call, it can be related to, for instance, population bottlenecks or demographic bottlenecks, whereas you have perhaps very few individuals in a population, so that at a given time and space, particular individuals reproduce more locally because they are there simply a little bit by chance. So this is, and then you will have the same type of scheme. This individual will lead to more individuals simply because of what we call drift. So neutral processes that lead to particular population enrichments in a given variant. Okay? So this looks simple, and it actually is, but we need to understand how this can impact our way to understand biological patterns. So evolution, as I said, this is a summary again, is a process that results from two major types of underlying mechanisms, the most important of which is what we call Darwinian evolution. And this process, or these processes, determine the way, the patterns of biological diversity that we see. So the species diversity, how species relate evolutionary to one another, the phylogenetic patterns that we see, and also how species distribute along a space and time that we talk about biogeography here. So these mechanisms imply or explain have the potential, the explanatory potential to explain the patterns that we see. So if you understand this, you understand many things about biology. However, very often we hear some assumptions or some affirmations that somehow translate a poor or a partial understanding of this type of causal mechanisms behind or underlying biological diversity. And this is what I will talk about today. Many of these processes or these misconceptions or misunderstandings or whatever you want to call it are due to actually to the way we think as humans. So many of them relate to what we could call teleological thinking, the idea that things evolve for a particular purpose. So there is a finality. Many of these misconceptions relate to the idea that we have, which is quite inherent, that things don't change. This is what some people call essentialism. So, for instance, species, they are units that don't change. Things have an essence. And most of them actually relate to our inevitable, in a sense, anthropocentric thinking, the way we think. I will come about these aspects with examples. And finally, I think that some particular misconceptions or doubtful assumptions come from, I would argue later, particular views that physics and chemistry have about the evolution of the universe. And this regards essentially to complexity, which is a delicate issue, and I will come back to this later. So some examples. That birds evolve wings to flight. So everything that I will quote here, put in quote, represents some of these misunderstandings. So that birds evolve wings to fly is one of these is a very good example or illustration of this teleological thinking, the idea that something, an organ, evolved just because there was a function that we needed to reach somehow. There was a finality here. But of course it doesn't work like this. If you understand the mechanisms that underlie how evolution works actually. And most evolution works by little steps. So before a wing, an organ that is used to fly, evolved, there are many intermediate steps that took a long time in which or during which you had an organ that was not able to perform the function flight. So people in evolution, we talk about adaptation, so little steps, little mutations that trigger little changes that perhaps improve some particular function. And actually how wings evolve, if you study that with time, for instance in birds and their ancestor, some kind of reptiles, is that there were some organ that progressively developed more longer bones, etc. And perhaps at the beginning it was just helping to walk, or help to keep the equilibrium, or help to jump a little bit further, but was not used to flight. Until at a given moment that organ was large enough to really start to fly. So this is, we also talk, evolutionary biologists talk also about exaptation, which is underlying this idea that before an organ like a wing evolved, there were many different steps with different selective forces that were selecting that particular organ to perform another function that was not flight. And at a given moment, a function flight could also be co-opted. We talk also, that's the process of exaptation, co-opt or take an organ that is performing another function to perform a new function. Okay? So when biologists say that birds evolve wings to fly, we tend to speak like this. But please understand this as a metaphor of purpose, because evolution is essentially purposeless. It means that during the evolution of the wings, all the functions, all the individuals that did not perform the function for which the intermediate or the older organ was used, they were eliminated by natural selection for whatever reason. And so this particular organ was what remained successful to perform all the intermediate functions that it had during the past. So this is very important, because we tend to speak like this, birds evolve wing to fly. This is a shortcut to identify, this is a metaphor actually, because evolution, as I say, is purposeless, essentially purposeless. This is an important thing. Many other misconceptions, or perhaps also the former one, can be understood in the framework or in the context or our own observational bias. That is only natural. I mean, we are material, we have this size, this body, and we have our senses that tell us one particular reality. So it is only normal that we have an observational bias, because we are like we are. A product of evolution, by the way. And of course, as many, it is only natural that we are anthropocentric in a sense. Every individual places itself or himself or herself in the center of the universe, and every species as well. So this is only natural. But our senses, the way we perceive reality, influences our way, or the way we interpret and we understand the natural world around us. So this is important. And actually, when we look around us, what we see is a huge biological diversity of animals and plants. We distinguish different kinds of morphology. We humans have been trying to class animals and plants, often in forms of trees, but anyway, for many, many centuries, we have classification schemes for those kinds of organisms. And when you go and look into species catalogs, you very often see numbers like these ones, that you have more than 1 million species of animals. Yes, Muriel, always calling attention. That's your biased perspective. So you go to catalogs of species and you see, okay, you have more than 1 million species of animals, and most of them, by the way, are insects. 1 million species of insects. So most of the biodiversity on Earth is insects. Or you have like something 270,000 species of plants. And then when you go to catalogs of species for bacteria and archaea, there are taxonomic books about this. Well, you count perhaps not even 10,000 bacterial species that are described. And so by reading these numbers, you will have the impression that most biological diversity lies in animals. And this is, of course, wrong, extremely wrong even. And this relates to our, the way that we perceive the natural world. So if I present you this series of portraits, so you will be able, perhaps some French more than others, you will distinguish all these different individuals. You will be able to put names under the faces. But if I show you this picture, maybe you will have already more difficulties in differentiating all the different individuals in this species, just because this is a distant human population, and the opposite is also true. Means that Asian populations will have more problems to differentiate Western type individuals. And if you go now to species that is more divergent, evolutionary, a distant cousin if you want, then you will even have more problems to differentiate individuals between them. And when it comes to bacteria, well, not only you are unable to differentiate individuals between them, but you are unable to differentiate different species between them. And this is actually because our perception of this kind of world is not performant. We are not adapted to observe the chemical differences that are the ones operating at this level. So our perception of the natural world is, of course, biased. And it is biased because of a problem of scale. There is a problem of phylogenetic scale. So the more distant you are, evolutionarily speaking, to another species, the more difficult it is for you to differentiate individuals from that species. There are problems of spatial scale. We are big and bacteria are small, and as I say, we are unable to, our organs, our senses are not able to differentiate the distinctive characteristics that are operating at this level. And finally, there is also a problem of temporal scale. This is a different kind of problem, but it's also related. And by that I mean that we as humans, because we have a given time length, we are unable to and somehow comprehend very long evolutionary or time scales. It's difficult for us to really grasp geological time scales, for instance. But that is important, because think just a moment about it. So this is representing a time scale since the Earth accreted some 4.6 billion years ago until present, and then you have the origin of life appearing. We don't know exactly when, but let's say around 4 billion years ago. And then you have at some point, perhaps around 2 billion years ago, the origin of eukaryotes, and then you have some 600 million years ago, the emergence of big multicellular animals and then plants. So things that are multicellular and have organized, more or less organized tissues. I'm not talking about colonies here, but just things that have organized tissues, and they appear really quite late. So if you now take into account this scale, you will see that most of Earth's evolution actually, or the planet history, has been occupied by microbial life, and that big multicellular organisms really spread like not yesterday, but the day before yesterday. Okay, so really very late. So this represents a huge evolutionary time where species diversified and microbes diversified, even if large things that we think are the most diverse appear much later. So they have much longer time to evolve and diversify. And this is actually one of the reasons why and this is another message that I want to convey here very strongly, this is one of the reasons why most biological diversity is microbial, and we are just appreciating this in the past 10, 20, 40 years at most. So this represents a tree of life as we more or less know it today. Karim mentioned just this yesterday, so we recognize now 3 major domains of organisms, 2 domains of prokaryotic organisms, which are morphologically simpler, but that have many different kinds of metabolic capabilities in terms of energy metabolism, energy transduction, and carbon metabolism. And each of these triangles represents perhaps thousands of sequences that are the thing that we use as proxy for differentiating microbe, and I will come back to this. And then a third domain, the domain of eukaryotes, that is actually a derived domain. So eukaryotes derive from symbiosis between archaea and bacteria. We know that now, but that's another story for another day. But underlying this, just most biological diversity, even within eukaryotes, where there is the domain where animals and plants lie, but also fungi, most of this diversity is again microbial. So multicellular organisms appear really having more or less organized tissues, and I was saying big organisms appear where the stars are. There are other ways of making multicellular organisms, but these are the most conspicuous ones, the ones that are bigger and with more different kinds of tissues and cells. And because, this is another comment that I'm going to do on this tree, because the positions of the stars are scattered in this tree, this means, in evolutionary terms, that multicellularity evolved independently several times from unicellular relatives, from unicellular ancestors. So this is another comment. So multicellularity is not something very weird, etc. It's something very easy to do. It has happened several times in the origin of life. Photosynthesis, oxygenic photosynthesis only appear once, so that's something very special. And eukaryotes only appear once. So this is for a general comment in this tree. I will comment more on this tree because one of the important things that I want you to understand, because many of you are not biologists, is why do we know? Because you could tell me how you know that this diversity is here. How can you know? And actually, because we cannot differentiate this diversity with our own eyes, because there is a problem of scale and everything. So how do we know? And actually, we know that thanks to molecular tools. And I want to say a few words about how we study microbial diversity today, just for you to understand what underlies the building of such a tree, which in this case it's a sketch, but represents what molecular trees tell us. So actually, studying microbes has been a problem for a long time because they are small. So the first thing that had to happen is that we developed tools to see them. And actually, it took a long time because we had to wait until the early 18th century to be able to see microbes using microscopes, very simple microscopes. So Anthony van Leeuwenhoek was among the first to describe the first microbes, possibly bacteria, and their movement using these very kind of simple microscopes. And then the evolution of this technology, different kinds of microscope getting better and better, allowed us to have increased details about this kind of life that was just open to our eyes only 2 to 3 centuries ago. And this allowed us to discover very important things. First, that all life is composed of cells. This is a cell theory, that all organisms are either cells, single cells, or composed of cells, multicellular organisms. So this is something very important. But also it allowed us to reconstruct the first so-called universal phylogenetic trees where you display relationships between species in the form of branch dichotomies based on morphology, even if morphology does not tell us a lot about the smallest microbes because they look very much the same. And then, even using more and more sophisticated microscopes, including the electron microscope, we could start to differentiate in different kinds of cell structures, and actually two. The prokaryotic cell structure, so like bacteria, like archaea, where you have the genetic material directly in a cytoplasm, and then the eukaryotic cell type that is on average more complex, having an endomembrane system that separates the genetic material from the rest of the cytoplasm where protein synthesis, etc., takes place. So in our way, or in our progress to study microbes, the first important thing was a technological invention. It was the microscope. And the second came with molecular biology, because cytology showed its limits, and once you have the morphology, you cannot go much further, and the ultrastructure, you cannot go much further. So molecular tools and molecular biology was somehow instrumental by the mid-20th century to announce the next revolution that has led us to actually be where we are here now, knowing that most biological diversity is microbial. So the idea that first DNA is the molecule that stores the genetic material in cells, and then what's its structure, and all the tools and knowledge actually that we derive from this in the following 50 years. And actually the development of these molecular tools was instrumental in solving a very important problem for microbiologists, which was exactly how to make a natural classification for microbes, for bacteria, because by the morphology you cannot establish a phylogenetic tree like you could do for animals, for instance, just by looking at the fossil record and how the anatomy has changed through time. You cannot do that with this kind of organisms. First, because the morphology is very similar all over. So this had actually represented a very important problem until the 1960s, and at that time, very important microbiologists like Roger Stanier, but also others, had somehow given up, had said, okay, it's impossible to establish a natural classification or a biological classification, a classification based on evolutionary relationships among bacteria, because we don't have the tools. But hopefully this has changed, and this changed precisely in the 1960s, thanks to the development of molecular phylogeny. So until now, until this molecular revolution, and I'm sorry it's too light, so you don't see that there are kind of tubes drawn here. what microbiologists had been doing was just to study the properties of microorganisms in culture. So you take your natural samples and then you can, what microbiologists call isolate microbes. That consists in diluting samples either by serial dilution or trying just to seed these microbes on solid media, solid agar media, to get colonies. And then the idea here is that you get colonies in the end if you dilute enough, in which all the cells are derived from one single cell. And then this means that you have a clonal population there, and that all the individuals belong to the same so-called species. And then once you have isolated bacteria, let's say, then you can understand how these bacteria react face to different culture media, and understand whether they are able to respire oxygen or sulfur or nitrate, or if they can produce that kind of metabolites or that other kind of metabolites. So these are phenotypic properties that for some time were used to establish a kind of numerical taxonomy, a kind of classification key that was based on metabolic properties, but that had nothing to do with evolutionary relationships. So as I said, things change a lot with molecular phylogeny, and molecular phylogeny is based actually on the capacity of some biological polymers to store information, and information about past history. So this idea was actually very well formulated by Emil Zuckerkandl and Linus Pauling in a very famous paper, Molecules as Documents of Evolutionary History. And here the idea is very simple. It is the idea that in biological macromolecules such as DNA or RNA, nucleic acids, in the sequence of bases, nucleotides of this molecule, but also in the sequence of amino acids in a protein, you can store evolutionary information. So the idea would be then that you can pick one molecule that is present in all living beings, from bacteria to elephants to plants, and then by comparing the sequence of nucleotides or amino acids in a given molecule, then you could compare all the organisms. So the idea would be to pick a molecule that is conserved in all living beings, and then you can compare the same character in all organisms. And by that, you could be able to reconstruct a truly universal phylogenetic tree. And those among you who are biologists know that this happened very well, and that we use that today for many, many different things. And actually, you have for sure heard about Carl Woese, who was the first to apply this idea of Zuckerkandl and Pauling to this very big problem of establishing a natural classification for microbes. And actually not only for microbes, but for the whole living beings, no? For everyone. And the molecule that he chose at the time, it was the ribosome of the small, the RNA of the small ribosomal subunit, which is very conserved. It means the ribosome is the machine to synthesize proteins, and we all need proteins, so this is a highly conserved machine. And actually, the important or the key elements of the ribosome are more particularly the RNAs. And so he picked one of these RNAs that was long enough to have enough characters to compare all organisms. So at the beginning, it was in the late 1970s, he picked, or kind of sequence, at the time it was catalogs of nucleotides, but anyway, kind of sequence this molecule for a variety of eukaryotes, including plants, yeast, humans, and then bacteria. And there were two important things. So this revolution implied two important things. One was one answer to the question, can we use molecules to reconstruct an evolutionary tree of organisms? And the answer is yes, we can. And here is the tree. And the idea is that more or less the length of the branches is proportional to evolutionary change, to the number of mutations. This is more or less true. Well, at this scale is true. And the second thing was something unexpected, and it was that he was analyzing our sequences from a variety of prokaryotes bacteria, what looked like bacteria, but he noticed that some of these weird bacteria that were living in extreme environments for some of them, high temperature or high salt or in anoxic conditions, then well, these organisms place apart from bacteria and form a distinct group. He called these organisms the archaebacteria. Now we call them archaea. He recalled these organisms archaea a bit later. Okay, so we discovered a new group of organisms by applying this type of very basic idea. So that is one thing. We can classify organisms and reconstruct past history based or exploiting this molecular information that is stored in DNA. But second, and this is another kind of revolution, or it's one of the outcomes of this molecular revolution, is that we can also apply this as a strategy, as a tool to study microbial diversity in natural environments, because microbiologists have known for a very long time that most microorganisms in a natural sample, don't grow in the lab. I mean, you can put more or less effort, you can get much less than 1% of the diversity growing on a plate, or maybe up to 10% if you put a lot of effort, but really a very tiny fraction is amenable to culture in the lab. This means that most microbial species, we don't have access in the lab to them. You cannot study them in the lab. So how to evaluate their diversity? Now we can, because we can use again this molecule, the ribosomal RNA gene, for instance, as a proxy to estimate the diversity that is present in natural environments. If we have the sequence of this marker, you can use this as ID card if you want, and then you will have access to the whole diversity that is present in a system. And this kind of approach we have been applying for the last several decades. This is how biologists have been doing. So at the beginning, what you do is that you have a given sample, can be soil, can be water, can be whatever you want, and then you extract the DNA from the community, and then you amplify from this DNA. You have many genes, of course, and that of the 18S or 16S in the case of bacteria, 18S in the case of eukaryotes, the rRNA gene, and then you can amplify. It means make many copies to have a lot of material. This gene using specific primers for bacteria, for archaea, for eukaryotes, or something universal if you want. So you can amplify the gene, and then the way people used to do this is to clone these genes and to actually this is a strategy to separate them, and then you sequence these individual genes that you have cloned, etc. And then you can compare these genes with all the genes that we now have in databases, and then you get the more similar organisms, and then you get organisms that you can use as reference in databases for which you have information, and you can reconstruct phylogenetic trees of your environmental sequences plus the sequences that may come from other environments, and also from cultures that are present in the database. And in this way, you can establish what microbiologists call operational taxonomic units. This is important. I'm going to talk more about this. And operational taxonomic units for microbiologists is our proxy for species. So We use this term because we don't know very well how to define species, but we can define operational taxonomic units. So it's something operational. And what we do, so an operational taxonomic unit for us is a group of sequences representing, of course, organisms that share between them more than a given threshold of identity, so that they are in the one very common value is 97 or 98% identity at the level of nucleotides here. So when you have two organisms that have sequences there are more than 98% identity, then you consider that they belong to the same OTU. You consider that they belong to the same species, microbial species. So that's an important concept. And by doing this over the years, okay, the first kind of studies date back to the early 90s, so a long time ago. You were possibly not even born. But then at that time, using this kind of approaches, people realized that you could discover new groups of organisms in the environment. The first kind of environments where this was done was the oceans. So you have groups of organisms like this SAR11 from the Sargasso Sea, which is now known as the Pelagibacter clay. These are ubiquitous bacteria in the ocean, very abundant, very important. But you have also new groups of archaea discovered in the oceans, including some archaea that are now known as the Tomarchaeota, this tag of Superphyllum, etc. Well, very interesting archaea discovered in the oceans. This is another story. But anyway, groups that we now know are very important, that are key, by the way, for the nitrogen cycle at the level of the Earth. And we discovered them by these molecular tools in the early 90s. So this is something important, actually, for many reasons. And then we have been accumulating sequences in databases, some of which are curated. This is one of such databases that keep these reference ribosomal RNA sequences for bacteria, but also other things as well as reference. And then this is just a review in 2014 about the accumulation of these sequences that represent microbial species in this particular database that only keeps sequences of a given length and that are of good quality, because there's a lot of crap out there. And so they keep an alignment of more than 5 million aligned sequences and longer sequences, etc. And the idea here is that with time, of course, there are many more sequences that are deposited in these databases. This corresponds to this release in 2012, I think, reviewed in this year. And with time, the number of OTUs also increases. And in 2012, when this database was released, this is important for you to understand the extent, the real extent of microbial diversity. Around 75% of the new entries, new sequences in this database corresponded to the species levels. Those 75 of each new sequence, so 100 new sequences, 75 new species, and about half of them new genera. So this is important. Genus is around 95% identity threshold for bacteria. So, and actually the trend goes on. So we have not yet seen a limit. And this is all tools. Now we have more, we have improved the way we sequence. So improve, I don't know if that's an improvement, but at least we can sequence more for less money. And now what people are doing these days, I don't know, some biologists between those, a lot of masking amplicon sequence. So here what you do, or people call also about metabarcoding. Metabarcoding, it means that you can amplify again a portion of this marker gene from DNA that you extract from the environment, like before. But now instead of cloning and do all the manual steps that you had to do previously, you just sequence. And you have, there are many sequencing techniques, but you can use this. These are the ones that are more favored now, but this will change maybe in a couple of years because this is changing all the time. And now you can generate billions of sequences in just a few hours. So, actually, of course, you cannot then analyze all the sequence by hand. You have to apply bioinformatic tools, so people try to remove errors because there's some error with these techniques of sequencing techniques, and that you have to remove to retain only high quality sequences. And then you make first some kind of OTU clustering. So you don't make phylogenetic trees before and then decide which is the OTU that you want to define, but you kind of use similarity tools to group sequences that resemble together, and you define then OTUs, and then you can pick a representative sequence from each of these OTUs and make phylogenetic trees that you can represent with this kind of star form, and to analyze the diversity that you have in a given sample. And this is revealing a huge amount of diversity, including new groups. But always in these three domains. We have not discovered new domains. That is not clear. In addition to these methods that rely on the sequencing of a gene, just one gene marker, you can also apply other techniques that biologists call omics, metagenomics, etc., that will target not only one gene but the whole genome. So you can analyze now, using metagenomic approaches, the genome of a community. You just extract the DNA and you sequence, and then you get everything mixed, of course, but you can look for particular genes and marker genes within this community. And then you can do all the fancy things like metatranscriptomics, so sequencing the transcripts, or even go to the proteomics and analyze the proteins of these communities, but this is more difficult, so people tend to do it less. Usually microbial communities are complex, so people try to compare or combine this kind of approach with approaches that target individual cells or subgroups of cells. But if you separate single cells from a microbial community, a single cell does not have enough DNA, so you have to pass by amplification procedures. So there are many ways how you can get single cells from communities. These are just examples, so this is just to show you how biologists work now. You can micromanipulate cells, for instance. You can use microfluidics to separate individual cells within droplets, or you can use more fancy machines like these flow cytometer machines that can separate individual cells based on the given fluorescence. You can use particular dyes or even the autofluorescence, and you can illuminate these individual cells that you pass through a capillary to sort them by inducing some kind of induced charge on the surface of these cells. So you can sort cells, and then you can amplify either the genome or the transcriptome, so the RNA or the DNA, and study the RNA and the DNA from individual cells. And by doing that, actually you have access to an even larger diversity. Why? Because many ribosomal RNA genes are divergent, and then you cannot target them by using primers that weren't used in the past, because some true universal primers don't exist, and some divergent organisms are not picked with common primers. So this has allowed to discover this kind of metagenomic tools, or single cell genomics tools have uncovered an even wider diversity. People talk about this microbial dark matter, and this corresponds to new lineages of organisms for which we don't have no information but the genome. We have information, or partial genomes in general. You have information about the genome of organisms that are living there out in the environment that you cannot cultivate in the lab. But we know that these are widespread in the tree of life, and this is even an even more recent view of the tree of life, where all these collapse things correspond to groups of microorganisms. So here you may have thousands of, in this particular case, is ribosomal proteins. And all the dots here corresponds to groups of organisms, candidate divisions, phyla, so big group of organisms for which, and this is only the bacteria, this is the archaea, eukaryotes are a little represented here. And all the red dots correspond to this kind of big phyla, candidate divisions, for which we don't have representative in cultures. So these organisms exist out there in nature, but the only information that we have is this gene marker. So there's a lot of diversity out there. Again, reinforcing the idea that most biological diversity on this planet, when you look into environments that can be more common like oceans, etc., or soils or fresh water, but also extreme environments, actually most of this diversity is microbial. Okay? And then coming back, so this was just an introduction, and then coming back to the misconception. No, but this is for you to understand how we analyze this and the real extent. I want you to grasp this extent of biological diversity. So when you read these numbers, actually 10,000, no way. I mean, this is, of course, a problem of bias. Nobody describes a species. You don't cultivate a lot of species in the lab because they escaped our way. So we have to use other tools. And actually, this is also a problem that relates to the species concept. I come back to this. Because traditionally, biologists in general, when studying animals and plants, and even some microbes, so you can see under the microscope some microbial eukaryotes that have interesting features, dinoflagellates, foraminiferans, diatoms. They have forms that have been used in the past to classify these microbes in different species and classes and etc. Using this morphological species concept, the idea that similar morphologies mean the same species. But we know that there are many problems here, particularly with microbes. And in particular, we have many cryptic species. So actually, most of the bacteria are a single cryptic species, or are a complexes of cryptic species. Things that are morphologically identical or very similar that you cannot easily distinguish, but when you go to the molecules, they are very different. So this is one such example. This is one microalga that is very common in oceans, Micromonas pusilla. It is small, it has a chloroplast, a nucleus, a mitochondrion, just one of each, it's enough. And so this is an electron micrograph of this kind of microalgae, so 1 micron, the size of a bacterium, very small. And then they are very common in oceans, and when you isolate them and make the sequence, you can have even the genome sequence. They are very different in terms of genetic information. So it means that there is a huge hidden diversity that we call cryptic diversity, cryptic species complexes, in this kind of organism. So morphological species concept is very limited. Then you have the ecological species concept. The species would be correspond to organisms occupying the same ecological niche. We don't know how to define a species, but a niche, it is even more difficult. So defining a species as a function of the niche it occupies is extremely difficult, because defining a niche, an ecological niche, is difficult, and recognizing it. What is the niche for a single bacterium in the ocean? It is what? It's not that easy. So it is the ecological function that it performs in terms of gaining energy. It is the ecological niche that it performs in terms of interaction with other species and how to measure that. That is a very extremely, it can work in theory. It is extremely complicated to use it in practice. Then we have the biological, the classical concept, interbreeding. If you cross with female and male, and you make a progeny, then you belong to the same species. Is it true? Well, if you do it for plants, you can cross many plants and have hybrids. And some people say, okay, you can have hybrids, but then they don't reproduce. Well, it's not true. It's not even true for animals, which is the only maybe group of organisms where this biological concept would work. And actually, you can cross tigers and lions and get a progeny that is viable and can reproduce. So it doesn't work even for animals. So what is left? The phylogenetic species concept. It means evolutionary relationships, things that are related, closely related, belong to the same species. Then the problem is where to put the threshold. And actually that is the only concept that can be applied to bacteria, for instance, because it's the only tool that will allow us to differentiate prokaryotic species. So we can define these OTUs, operational taxonomic units, and set a threshold, say 97% of nucleotide identity at the level of this particular gene. And now, if you take this as a concept for prokaryotic species and you apply it to animals, what do you see? Well, you see that all primates belong to the same species, 99% identity. So we are the same species. But also mice, so we are the same species. And you have to go as far as the ornithorync, yes, to fall to a 90% identity threshold at this level. See, this is the genus, so we are the same genus as the ornithorync now. So actually almost all mammals would be a single prokaryotic species. So if you use this definition and you apply it to mammals, you can see that prokaryotes, microbes, are really much more diverse. So this is something that we need to think about it. And that's for this part. I come now to a very different story, and it is a problem of complexity that I mentioned at the beginning. This is complex. So I mentioned at the beginning that part of these misunderstandings come from the idea that there is some kind of law imposing an increasing complexity to the universe. And many physicists and chemists, not all, but many, will tell you that there must be some kind of law that we have not yet discovered that applies to biology and that imposes this kind of increasing complexity. And this comes from an observation in fact, and it's the idea that, of course, when you look at the universe out there and you see and to the chemistry of the universe, you see that from hydrogen you have nucleosynthesis, and you have heavier and heavier atoms, so you increase the complexity, the atomic complexity. And then you have organic molecules that appear to form. You have water, you have methane, you have organic chemistry that set up, and then you see some kind of chemical evolution. By chemical evolution means an increase in the diversity of molecules in the universe. And so it is only tempting to say, okay, well, there is a trend, an increase in complexity. And this applies also to biology. So actually, you can draw this kind of sketch, and you put complexity and time, and there is an increase in complexity from the beginning of the universe to life, and it's always increasing. And you could see it at the level of individuals, but perhaps not only because some individuals are reduced, I will come back to reduction later, but in terms of the ecosystem. So you get always an increased number of interactions, and that can also be interpreted as increased complexity as a whole. So many people will tell you this, there's an increasing complexity. And maybe, okay, so actually this is a view that could apply to life or not. It means that you have complexity that increases in the universe, and maybe life we don't see it, but there is a bar here in green or something like that. Imagine you define that this threshold means life, okay? You draw a line here and say, okay, more or less here I see animals there, okay, life appear here in this line of increasing complexity. Okay, as I said, it can be viewed at the level of units, living units or not. And this is very often related, you will hear people telling this, to the idea that complexity improves function. Okay, another misconception, but anyway. So the idea that, of course, if you go, and very often many authors use this type of how to say, technological analogies to explain this. So if you increase the complexity of this, you get an airplane. So we are always with the analogy of flight here, and then, okay, so you could put a bird and something before. But is this always true? Actually, no. Not even in technology. So you can use the same analogy, and you can build very weird and complex some kind of airplanes that actually don't fly well or don't fly at all. And you have also low-cost companies. They reduce many functions there, and they still fly, and they are cheaper, and they are performing well. So, well, maybe it's not always true, even in technological analogies. So does complexity always increase, including in biology? Of course, at the beginning, it was the case. I mean, before life appeared and biochemistry appeared, there must have had an increase in complexity, the organic chemistry, etc., variety. By complexity, I mean diversity of organic molecules, and also the minerals, etc., all those, the complexity that you can imagine in the inorganic and organic world, but abiotic, prior to life, which is set a little bit more or less there. But at a given point, perhaps what happened is this, that there was, of course, an increase in complexity, but there was perhaps some kind of phase transition, and we are assisting to a kind of asymptote where complexity is no longer increasing on average. It means that, of course, there are things that appear and that are new, but are things that also evolve by reduction. And I'm explaining better this because, and this could apply also to ecosystems, by the way. I mean, if you talk to ecologists today studying ecosystems, they will tell you, okay, all the niches, if biology can't colonize a niche, it is already there. There are no free niches. So you have to remove someone from a niche, an ecological niche, to be able to get to make the space for another one. So it is perhaps we are assisting to what ecologists call, and physicists should call that as well, a stationary phase for interactions within ecosystems, and it has to happen something else to remove the space or to create new space for new interactions. So the interactions can get even more complex, that's for sure, but they can also get simpler. And it is the same for biology. And actually, reductive evolution is extremely important. So, in biology, the number of genes is very often used as a proxy of complexity. You have more genes, you're more complex. It's a relationship that can be reasonably supported. So here you have a diagram showing different kinds of genome size ranges for different groups of organisms. I don't think that you read it from behind, but anyway, you have here bacteria and archaea. These are the genome size ranges, maybe 1, 2 megabases for not very big genomes. The genomes are compact. And then you have many other groups of organisms with more or less big genomes. Mammals are here. And you have, by the way, things that have much larger genomes, that may have much larger genomes than mammals, for instance, including many protozoa. So these are unicellular eukaryotes, unicellular microbes. So many unicellular microbes have genomes of higher genome sizes than our own genome size. So think about it. But actually we know that many organisms reduce the genomes. And this happens all over the tree, along all these groups of organisms. So that one clear example is that of parasites and symbionts. We know it's for sure that when you have obligate symbionts and parasites, these organisms get rid of many functions that they don't need. They lose the genes. So they have reduced genomes, and well, that are much smaller than free-living organisms. And you have this in all known groups of organisms, where you have free-living and symbionts or parasites. And there are even many people who think that reductive evolution has been a dominant element along biological evolution, and that most biological evolution has been driven by gene loss, except that at some points there is pulses of gene gain via, for instance, whole genome duplications. We know that has happened, for instance, at the onset of yeast or some other organisms, that you have gene duplications and then you can do many things with these genes that are duplicated. You keep one copy to do the thing that they have to do, and the other copy they can evolve freely and lead to new functions. And then with time, many of these genes are getting lost, or horizontal gene transfer. That is the other source of gene gain that an organism can have. And then with time, you lose many genes, especially in microbes, because having genes that you don't use is costly. So if you live in an environment that is nutrient limited, and you don't really need the function, you just lose the gene. And then if you need the gene again, you can gain it from a neighbor, because horizontal gene transfer is quite widespread, especially among prokaryotes. And you know that very well when you think about antibiotic resistance and how it is spreading. This is a lot by horizontal gene transfer. So if something is really needed, you will import it from your neighbor. Another misconception that is very general: simple means primitive. Well, a counterexample is that of Microsporidia. These are, again, it's an example with parasites. Microsporidia are very simple parasites, eukaryotic parasites, have a very simple structure. They look like bacteria practically. They have a very long structure. It is a polar tube that they use for infection, and they don't have mitochondria. Mitochondria is the organelles that we use for oxygen respiration. Actually, they have very reduced mitochondria again. So at the beginning, when we started to make molecular phylogenies for these kind of organisms, for eukaryotes, Microsporidia appeared as the first emerging branch in the eukaryotic tree, and together with other organisms that also appeared to lack mitochondria. And so they were thought to be primitive eukaryotes. I will come back to this word again, and form a clade that some people call archezoa. Again, the idea of ancestral or primitive, that would have predated the acquisition of mitochondria by the ancestor of the rest of eukaryotes. So we actually know now that this view is wrong, because of two reasons. One, these Microsporidia were placed at the base of the eukaryotic tree by an artifact of phylogenetic reconstruction that is called long branch attraction, for people who know about this. And also because now we know that these organisms have actually had mitochondria, and they still have reduced mitochondria, but they have lost most of the genes, and only some genes remain in the nucleus. And when you do phylogenetic trees today that are using better models of sequence evolution, and you use more reference sequences, actually these microsporidia come close to fungi, where they belong. So these are reduced organisms that are not primitive, that are simple, but they are simple by reduction because they have lost many functions and many genes that they don't need because they get many things from the hosts. This idea that simple means primitive underlies even some older ideas for the origin of life, and in particular the idea that viruses, because they are simple, essentially viruses are correspond to a genetic polymer, can be DNA, can be RNA, that is in its dispersive form surrounded by a protein capsid. It can be also enveloped by a lipid coat that it comes from the host. But anyway, so very simple entities. And then some people, including Hermann Müller, who coined the term gene, equal gene to virus, and said that the first living organisms may have been viruses just because they were simple, and he was equaling or comparing gene to virus at that time. It was in the late 1920s. And even some people like John Haldane sustained at that time that life might remain in this state, viral state, for many millions of years before really the first cells appeared. Well, this was highly criticized at the time because precisely this idea that simple means primitive is wrong. You can have simple things that are primitive, but many simple things are simply reduced forms or escape forms. I'm not going to enter into the debate of what viruses are, but it is for sure that viruses have not predated replicators or cells, things that can support the replication, because they are obligate molecular parasites. And there is another thing for sure, because viruses have very different families with different kinds of genetic material, DNA or RNA, simple, single-stranded, double-stranded, positive, negative. They have multiple origins, so we don't know exactly when and how they have originated, perhaps escaped from cells, perhaps other things, but they cannot have predated and originated life, because they depend on replicators already. Anyway, and even Haldane recognized a little bit later that actually most evolutionary change has been degenerative, looking to parasites. Okay, to comment on this. And actually you can have unicellular organisms that are quite complex. One example here, and it's I think it's one of the most amazing examples of complexity that I know of. It is one kind of dinoflagellate, so these are microscopic unicellular algae that has an eye. Actually, its convergence is it's independent origin of the eye, if you want. And the eye is formed by a chloroplast, and it has a cornea and a lens and a retinal body. This retinal body is a transformed chloroplast with pigments, and it performs the function that our own eyes perform, so it can see this organism. And some of these species belonging to this group of dinoflagellates can even propulse themselves with kind of piston when they see a prey to catch it. So they see and they go and catch the prey in one single cell, very complex. So this argues against the idea that bacteria or unicellular organisms are simple necessarily. And of course, that argues also against the idea that bacteria and unicellular organisms in general are less evolved than animals and plants. This is very strongly anchored. You still see in many textbooks, it's a pity, it's like the cyanobacteria, cyanobacteria that you still see in textbooks. You see this 5 kingdom classification of Whittaker, so 5 kingdoms, so the Monera, the bacteria, unicellular organisms that are of course placed at the bottom. They are simple, they are primitive, they are old, all the same at the base. And then the protists, so unicellular eukaryotes, these are like this anoflagellate that I just showed you, that are a little bit on top. We are eukaryotes as well, so I think that there is a little bit of eukaryocentrism here. Okay? And then on top, of course, you have fungi, you have plants and animals, and on top should be man. Okay? So this is kind of reminiscent of this Aristotelian scale of nature or chain of being, where you have the rocks, so the mineral world, the flames, okay, plants, beasts, and humans on top, and above humans you have angels and God and other fancy creatures. Okay, this is a little bit the same. And this relates to a given idea of increasing complexity, and we are on top, of course. And the things that we idealize are even on top of us. So things have changed. That is what I have tried to convey here. So from this view, that is not that old actually, and you still see in textbooks before the 1980s, where you have bacteria at the bottom, unicellular eukaryotes, and these 3 big groups of organisms, the thing has changed. And now what we have, sorry, it's not very clear here, the lights are color, but actually you have 3 domains of organisms, the bacteria, the archaea, and these 3 large groups here, fungi, animals, and plants, are just these tiny branches within eukaryotes, which are mostly microbes, and these are kind of not extremely diverse if you compare them to the diversity of prokaryotic organisms. So this is another way to see at the same kind of scheme. So this should represent the tree of life. The colors are light, but you imagine there are 3 domains. One of them is derived actually, merging or symbiosis between archaea and bacteria. And then if you zoom in a particular tip of the one particular branch, you will find humans and primates here. It's a tiny branch. And of course, if you pick up this branch and you take it back to the last common ancestor of all organisms after life appeared, of course, life appeared somewhere after the prebiotic world, some 4 billion years ago. Of course, there is complexification to a last common ancestor, because at the beginning you need some increasing complexity to make some kind of cell. And then to get to the state of a last common ancestor, the last common ancestor we'll see tomorrow is something already quite complex. And you may have had plenty of extinctions, many origins of life that have disappeared, for which perhaps some of them have contributed through horizontal gene transfer to this main line from which we derive. And of course, if you take this way back and you start it again, just going to humans, you see an increasing morphological complexity. But if you take any of our contemporary organisms, so pick a microbe here, and you do the same, and you get back, well, you don't see necessarily a lot of increasing morphological complexity. But that does not mean that these organisms have not evolved, because evolution is an ongoing process. So species are not fixed entities. They mutate, they evolve all the time. It is impossible to maintain biodiversity unchanged. And this is something, I say, this is something that conservation biologists need to learn, because you cannot preserve a species as it is, because species evolve. Actually, they are evolving entities. And so the idea that bacteria are old, that they appear very early and never change, is of course false. But we have problems to identify how bacteria have changed and evolved. But you can see that there are many species that are of bacteria that have evolved. Think about, for instance, our own microbiome. Before we were here, the bacteria that colonized us were not here. They were old relatives, different relatives, but the ones that are adapted to our gut, for instance, well, they appear with us or co-evolve with us. But also the bacteria and other organisms, yeast, that we use to make bread or wine, etc. Okay? They have evolved, and they are still evolving in companies. Another thing, and not only in companies, in nature as well, there was a kind of joke. Another thing, on the same line, living fossils don't exist. So please banish that word as higher eukaryotes, higher or primitive and higher, or that. Okay, all living organisms now, we are equally evolved because we have evolved along the same period of time. Okay, we are evolving. Different rates maybe, but we are evolved for the same amount of time. There is not a frozen bacterium, there is not a living fossil. So the coelacanth, many people say, okay, a living fossil, why? Because actually many representatives of the same big lineage were first known from the fossil record before it was discovered. But they are now extinct. And it was a surprise to discover this organism. But this organism is not old, it's living today. And actually, it has evolved. Because if you compare some morphological structure, the skeleton, but also the plates, etc., and you compare it with the other known fossil members in the family, well, they have important morphological differences for the experts. And actually, there is also a way of looking at this into phylogenetic trees. Of course, you can draw a tree with different organisms here, vertebrates, and put mice and humans here on top. And if you draw this kind of tree, it looks like Latimeria, which is the coelacanth, the genus of the coelacanth. It's branching very early. But if you remove these organisms and now you just leave Latimeria and Homo, you will just see the differences that are present between the two. And if you could have molecular sequences for these organisms, what you would see is perhaps something like that. You could draw a tree like this. So actually what is important is the difference in nucleotides between Homo and Latimeria, but you could draw it this way or this way, and you could induce another interpretation that is of course false. In this case, you could say that Homo is more primitive because it branches early. It's not true. They are different. They are half diverged, and they have so many differences. Anyway, more or less final comment about the limits of actualism, and this is actually insisting on something that Karim mentioned yesterday. This is the idea that the present is the key to the past, and it works to a certain extent, especially in geology. So you can interpret by looking to how rivers shape the landscape today. You can interpret all structures and say, okay, this was a canyon, there was a river, etc. So there is some power to this. But actualism has also limitations. So another example of actualism that works, and we heard about this before, is you have some structures in these beaches, and then you can ripple marks, and then you can see that fossilized some 2.7 billion years ago, and you can say, okay, this might be a landscape that was by the beach, etc. So you can make some inferences. But does it always work? Of course. And there is also the idea that functions in the past were the same as today. Okay? So the processes were the same. Were they really the same? So these are past fossil stromatolites, rocks produced by microbes, and this is based on the idea that we still see some of these communities today. So we know that there are some microbial communities that can induce precipitation and then get fossilized in the fossil record. Does this mean that the organisms that were living at that time were doing exactly the same? Well, that is more difficult to say. And actually, perhaps the oldest, it might have been the case for these stromatolites 2.7 billion years ago, when perhaps oxygenic photosynthesis might have been there because oxygen started to accumulate a bit later, but we don't even know. But the oldest ones, well, we don't know. Perhaps they were just anoxygenic photosynthesizing bacteria. And again, this is actualism. This is based on the idea that these laminations are a consequence of a response to light, okay, and a stratification by light. And this is also a hypothesis based on today's knowledge, but you can question that. Is there another way to make the same kind of structures? Well, to some extent, yes, to other extent, no. And again, by applying this kind of actualism, you could say that, of course, because this resembles this, which is the famous microfossil by Schoft, interpreted long ago as a cyanobacterial filament 3.5 billion years ago from this apex chert in Australia. Well, for some time, and this is still in textbooks, so this is important because like the 5 kingdom classification, this still in textbooks saying these were cyanobacteria, but we now know, and actually this was a big debate in the early 2000s when Brasher reanalyzed the same fossils that are in London and identified this one but many other, and still started to say, okay, well, this is weird, this does not, perhaps these differences in diameter, this doesn't look like quite biological, there are some ramifications that are weird, etc. Are we truly in front of microfossils, or are we in front of things that look like life but are not life? And this is the problem of biomorphs. Things that can be generated abiotically, abiogenically, by the presence of particular chemical conditions. You could trap organic matter that is produced in hydrothermal vents, for instance, hydrothermal synthesis, Fischer-Tropsch reactions, things like those. So you could trap in this kind of mineral structures, this organic matter, and you could make things that look like fossils, microfossils. So, question mark. And again, another example, very recent, and we heard about this yesterday as well, these new, the Earth's earliest fossils, these hematite tubes that appear 3.7 billion years ago, because they look like some tubes produced by some microbes, like there are iron oxidizers or similar kinds of microbes. Well, first, the size is not the same, 1 micron, 100 microns, so the scale. And then, can you do these kind of things by abiotic means? We don't know. People need to try. My guess is that this is very thin as an argument, just by this kind of resemblance, without taking into account the scale. So some people call these dubio fossils, Martin van Kranendonk included. So this shows a little bit the limits of actualism. And of course, the organisms were not the same because they have evolved. These people talk about present-day genera. They mention Microbium, etc. Well, it's 4 billion years of evolution between these organisms, if they existed at that time. And if these were microfossils, and the ones that you see today in deep sea vents and other places, you have these kind of microbes doing these kind of tubes. But they were not the same, if they ever existed at that time. And so this points to the limits of actualism, and by extension to the limits of analogy. Analogy is very powerful and very useful when you communicate science to the public. But you need also to convey the message that an analogy is actually nothing that it can help to understand things and to translate a message, but it does not have explanatory power. So you cannot explain things by analogy. That is not scientific. You can. It could work if the same underlying processes were explaining a phenomenon. But if the underlying process is not underlying the phenomenon that you see, then it doesn't work. So analogy is good to communicate, but science shouldn't work by analogy. Okay? Because airplanes are not birds, and these are not birds. By the way, think about it. And a final word, and this I just added this slide because yesterday we heard a little bit about panspermia, and I thought that I would drop a word about this. What about panspermia? Panspermia means, by etymology, that there are seeds everywhere. And you see, you go to Wikipedia, I went to Wikipedia, there are many kinds of panspermia. People classify these in many things. People talk even about soft panspermia for the organic compounds, where we know that organic compounds form everywhere, but it doesn't necessarily mean that they, of course they can travel, these compounds, but they don't mean that they come from elsewhere all the time. You can also talk about lithopanspermia, we had about this yesterday, okay, these kind of things. So this is the idea that life moves around the universe freely. But actually, there's one thing. One thing is local spermia, I would tend to call that local spermia, interplanetary life transfer. It is unlikely. There are many things during this travel, but it is possible. Imagine a meteorite coming from Mars, but well, it's difficult anyway, because you have to spend some time out there. There is cosmic rays, there are a lot of challenges. Then you have to enter the atmosphere and survive the impact, etc. So a little bit of complications if it ever was life on Mars somewhere some time ago. But you cannot really seriously imagine life transfer from another planetary system to this one. So I think this is much more complicated. Okay, you could imagine perhaps Mars or an asteroid, something. But it's unlikely, but possible. We know some organisms can stand some conditions, even if yesterday we were mentioning organisms like Deinococcus radiodurans who can stand radiation, etc. But it can stand radiation when it grows in the lab, but not in a meteorite. I'm not sure that the experiment has been done, because Deinococcus, in order to resist, needs to have an active metabolism to repair. If it's sleeping, I don't think that the metabolism can cope with it. So that's another problem. But okay, let's say you could imagine that under certain conditions, why not? But panspermia does not mean this. Panspermia means, or originally and etymologically, means the transfer of life all over the place. And panspermia is not a scientific theory for the origin of life. It doesn't explain anything. It pushes the origin of life always a little bit farther. So to a point where there is no origin of life. And then you have a way or a door for things like this cosmic panspermia, which is a new form of vitalism, the idea that organic matter and life cannot come from abiotic things. So that is always produced by something or by someone or by some form of creator. So I think that we need to be very aware of this, essentially because it is not, panspermia is not a scientific theory for the origin of life. And some people use panspermia to refer to local spermia or interplanetary life transfer. That could work, but I think that we need to to change the name, because people on this arena, in this cosmic panspermia, some kind of fuzzy thing, they are using arguments by scientists saying, hey, panspermia is possible, and then because local spermia, they say panspermia is possible, other things are possible, and we don't want to caution this, we don't know to support this as scientists looking for natural causes of evolution and origin of life. So with that I'm done. Just remind you some take-home messages. Most biological diversity lies in the microbial world. I hope I have convinced you. But otherwise, if not, you can come to the lab and work on that. Except in the Lord, because maybe, no, that's a private joke. Complexity does not necessarily increase. Of course, it increases along biology, etc., but not necessarily. Reductive evolution is also very important. Simple does not, again, necessarily means primitive. Bacteria are not less evolved than animals or plants. If anything, they evolve faster. Living fossils do not exist. Species are not fixed entities. Evolution is ongoing. We cannot prevent it. Actualism and analogy have very limited value in science. They have a value, but they need to be used with caution. And panspermia is not a scientific theory on the origin of life. Thank you very much. That's okay.